Hi students, today we are going to discuss about Topper IIT-NEET Foundation. That Topper IIT-NEET Foundation we are going to discuss in 9th class biology. In that what is the second lesson? Cell. Okay, we are already discussed about food resources and in animals like different processes which are helpful for the animals during the searching of the food material and acquirement of the food material. So what are the mechanisms like mixed farming, dairy farming, and pisciculture, apiculture, like different processes we are already discussed in food resources and animals. Then coming to the next topic, that is about cell. What is meant by cell, and what are the components which are present in the cell, and what is the ultra structure of cell in plant and animals. Means if you observe the plant cell with the animal cell, there are some of the different characteristic features are also there in between the plants and animals also. So what are the differences between these plant cells and animal cells, and how we can we can define the cell, and what are the theories and what are the cell organelles which are present in the cell. So now we are going to discuss about that. Okay, so today we are going first one, some of the scientists according to the cell. There are some of the scientists which are working on the this cell. So there are from the first 16 century onwards, there are more number of the scientists, they are working on the cell observation and what are the cell organelles are present, and during the observation how do they found that cell, what is the mechanism behind the cell discovery. So these are all today we are going to discuss. So first of all, one of the scientists called as Johnson and Johnson. Francis Johnson and Eukaryotic Johnson. He is one of the scientists first time discovered the compound microscope. Because of the invention of the compound microscope only, we can observe different things which are surrounding on us, which are not visible with our eyes. Means for our eyes, up to some proximity only we can observe. Means our eyes having lower resolution when compared to this lens or microscope. Okay, so what are the which things we can't observe with the help of our eyes, we can see with the help of the microscope. So the discovery of this compound microscope was first time discovered by this Eukaryotic Johnson. So because of this compound microscope only, after that one of the scientists called as Robert Hooke observed the cells in the microscope in the plant bark slices. Okay, for example, if you see the outer layer of the plant, that is called bark. So if you take in that bark and you are making some small slices from that bark region, then we can observe a lot of hexagonal cells inside the, like how they will be looking like, means honeycomb like structures. Means whenever you are observing the honey hive, there are some hexagonal cells will be observed inside the structure. So like this hexagonal structures. So like these hexagonal cells like structures were observed by Robert Hooke whenever he can observe the slices of the bark which is obtained from the oak plant. So then later he observed the structures and he named them as cellulae. What is that name? Cellulae. Afterwards, from the cellulae, in the Latin word, the cell is nothing but a little room. so little room. So means he observed some small little room like structures in the plant body. So that's why he named again. He later named the cellulae as cell. What is that name? Cell. So like that, Robert Hooke first time observed the cell in the bark slices of the plant. Okay, then coming to the another important scientist called as Antoine van Leeuwenhoek. So he is one of the microbiologist. So without knowing the microbiology term only, he observed lot of different small things which can't see with our microscope. Means for example, any dirty water or any pond water or the tissues which are in between our teeth or the gums of the teeth and whatever the RBC cells which are present inside of our body and what are the cells which are present on our skin. These are all are observed by using the different variety of the lenses and they in that he observed some moving creatures in that nature. So that moving creatures later they are named as some bacteria. What are they? Bacteria. Like based on the that they are moving in nature but very small in nature, they can't see with our naked eye. So discovery of so many different organisms in the nature in different sources is found a brick stone to the foundation of microbiology. Means the father of classical microbiology, we can say him, Antoine van Leeuwenhoek. After that, another scientist called as Noel and Ruska. Who is that scientist? Noel and Ruska. He discovered that one of the microscope called as electron microscope. Electron microscope. This compound microscope is having the approximately 5000 to 1000 magnification. Means the resolution capacity of this compound microscope is approximately 5000 to 1 lakh. But this Noel and Ruska discovered this electron microscope. It is approximately 1 lakh to the 10 lakhs maximum resolution will be given by this electron microscope. Because of the discovery of this electron microscope only, later our scientists discover so many number of cell organelles which are present inside the cell. Means inside the cell there are so many different varieties of cell organelles are there, like mitochondria, ribosomes, endoplasmic reticulum, lysosomes, peroxisomes, glyoxisomes, and different cell organelles are present inside the cell. So that was observed and discovered later by discovery of this electron microscope. Okay, and another some of the scientists called as one of the scientists called as Schleiden and another scientist called as Schwann. So Schleiden is one of the German botanist and Schwann is belongs to the German zoologist. They both are he is working on plants, he is working on animals. Both are combinedly working on different plants and animals and observed that there are so many number of cells and so many number of organisms in both plants and animals and proposed a theory called as cell theory. Okay, what is that theory? Cell theory. So the cell theory is nowadays also we are using the cell theory postulations. So during the cell theory they proposed some of the postulates. So what are that postulates? Means every organism's body is basically made with cell. What is that part? Cell. Means for example, if you see currency notes, So in our India, the currency note, even lower currency also we can measure with 1 rupee. Rupee means, for example, if you see 1 rupee, 5 rupees, 10 rupees, means in every end of the rupees we can mention 1 rupees. Okay, rupees word, like that, 10,000 rupees, 1 lakh rupees, 1 crore rupees, 1 million rupees, like rupee we will add. Like that, in our body, in the living organisms also, our body is made up of basically with cell. And what is the second point means, cell is the structural and functional unit of every living organism. Means in every living organism which is present on the earth or nature, that will be contain cell. That is the structural, and it is applicable for structure, and it is also applicable for functional body in the organisms. So these two are mentioned by this cell theory. Later that, but this cell theory can't explain that how the new cells are arising in the living organisms. Means how the production of new cells will be arising in the organisms. That is failed to explain by this cell theory, which was proposed by Schleiden and Schwann. After that, another important scientist called as Rudolf Virchow. Who is that scientist? Virchow, Rudolf Virchow. So he proposed that cells are new cells in the organism are produced from the already existing cells, means from pre-existing cells. New cells are arising from pre-existing cells, which are proposed, means this is also called as omnicellular e-cellular. Omnicellular e-cellular means cells are produced from again cells. So previous cells are produces new cells. For example, if any damage is formed on our body, so on surrounding that part, there are some old cells are there. These old cells are undergoes a division process. During that division process, that cells are producing new cells. So that is called as cell lineage theory, or it is also called as omnicellular e-cellular. Means new cells are arising from pre-existing cells, which was proposed by who is that scientist? Rudolf Virchow. So after proposal of this Virchow cell lineage theory, again the scientists changed that cell theory, okay, and change some of the postulates which are present in that cell theory. Because if you see the microorganisms, in microorganisms, some of the, for example, if you take viruses, so viruses are containing only the outer layer, that is called as proteinaceous layer, and inside some DNA or RNA as the genetic material will be there. So means here the virus is a nucleoprotein structure. It's not having any cell or any cell organelles in this body, but it is living in nature. How it is living means the viruses are acellular organisms, and whenever these viruses are present outside of the environment, they are behaves like non-living things. But whenever they are enter into any organism's body, then they will behaves like living organisms. So both properties are there for these viruses. So one is they are acts as in non-living things whenever they are present on outside of the environment, and they are acts as living things whenever they are present in the living organism's body. Means the living, it is not having any cell organelles or replicative machinery to replicate or to reproduce their body cells. But whenever it is present in a living organism's body, then it uses some of the machinery which is present in that host body, and it reproduces and producing some new cells from many number of the cells. So that's why there will be some exceptional cases are there for this cell theory application. So that's why for they are again changed, modified the cell theory into every organism's body is made up of cells or the products of the cells. Means the cells are containing some proteins, carbohydrates, lipids, and some DNA or RNA as the genetic material. So these products are also made present in this cell. So that's why every organism's body is made with cell or products of the cell. And in living organisms body, the cell is the structural and functional unit, and the older new cells are arising from the already pre-existing cells. So this is called as cell theory, which was proposed by, who is that scientist? Schleiden and Schwann. Alfonso Carti. So he discovered the living matter of the cells. Means, for example, if you see one cell structure, in this cell structure there will be one membranous like structure will be there surrounding by this cell, and inside that there will be some genetic material containing nuclear membrane, nucleus will be there. So surrounding by this there will be some liquid material will be present in this cell. Liquid material will be present. So this nucleus and this protoplasm, this cytoplasm, both are considered as protoplasm. So this protoplasm containing different cell organelles are responsible for the function, living nature of this cell. Means inside the cell, the liquid material acts as a living matter in the organism. So that was discovered by this Alfonso Carti. And another important scientist called Fontana. Who is that scientist? Fontana. So this Fontana observed the nucleus first time the nucleus in the skin cells of eel. Skin cells of eel. Eel means one of the fish-like structure. Means something like this structure will be there. So this long body with contain, but it is containing some fins on dorsal and ventral side of this body. So this is called as eel. Means long body with dorsal and ventral fins, and it is present in water. Means fresh water or that salt water, oceanic water. This eel is present, and it is whenever you are observing this eel from upper side, you are observing means the lower body parts will also appear on the body. Means the body is looking like somewhat transparent in nature. The body is looking like somewhat transparent in nature. So in this skin of by taking this skin of this eel, and he observed that the nucleus in the first time. Who is that scientist? Fontana. Fontana observed the first time nucleus in the skin cells of eel structure. After that, later one of the important scientists called as Robert Brown, he discovered the nucleus which is present in all the living organisms in the centrally located cell. Means every living organism's body, the centrally there is some central molecule will be there, central cell organelle will be there. So that central cell organelle is responsible for inheritance of the characteristics because of the material which is present in the inside this central cell organelle. So this nucleus was first time observed in Tradescantia plant by this Robert Brown. In which plant he observed that? trade scantia. Trade scantia. In this plant, he observed the first time the nucleus. So in skin cells of eel, Fontana observed the first time nucleus. After that, Robert Brown in trade scantia first time observed the nucleus, which is centrally located, and the inside the material is responsible for the what is that inheritance of the characteristics. And after that, after this Alfonso Carti, another important scientist called as Purkinje. Who is that scientist? Purkinje. He first time discovered the living matter of the cell. So first Alfonso Carti observed that living matter in the cells. After that, later it was also observed by this Purkinje. So and Purkinje and one another important Purkinje, and he termed that living matter which is present inside the cell as the protoplasm. So in between them, one of the scientists called as Dujardin. Who is that scientist? Dujardin. He also observed the living matter which is present inside the cell, and the living matter inside the cell he termed as sarcode. What is the term? Sarcode. So Dujardin named the living matter which is present in the cell as what is that? Sarcode. And one of the term Purkinje also observed that same living matter, and he termed that protoplasm for that living matter. Okay, and another so these are some of the basic steps towards the cell biology. Means this period is said to be classical period of cell biology. So these are the basic foundation steps for after advanced cell biology. So this is about some of the discoveries. Then coming to the size of the cells. So the size of the cells is also differ from one organism to the another organism. So means some of the organisms are containing 0.3 microns in length, and some of the organisms are containing centimeters length also. For example, 15 centimeter length also. Means here the cell is 0.3 to 0.5 microns in diameter also. The cell diameter will be measures. So the cell measuring about 0.3 to 0.5 microns in diameter, and in some of the cells 15 centimeters diameter is also present. And you can see the cells containing, for example, if you see the bacteria are measured for about 0.1 approximately 0.3 to 0.5 microns in diameter. For example, if you see the higher organisms like in our body, the cell diameter is approximately from centimeters they will be there. So in our body, what is the highest length means the longest cells which are present in our body means the nervous cell is contains the longest cell in our body. And in the nature, the largest cell which is present in our nature is ostrich egg is considered as the largest cell. And in our body also lot of cells are there. Their size is also differ from one organs to the different organs. And in between these bacteria and viruses, there is another organism that is called as mycoplasma, or they are also Mycoplasma galli septicum or Mycoplasma PPLOS, pleuropneumonio-like organisms. These PPLOS are measuring for about 0.1 microns in diameter. How much diameter will be there? 0.1 microns in diameter will be there. So this is the lower diameter which is present in Mycoplasma. It is a bacterium. After that, in some of the bacteria, it measures, the cell is measures for about 0.3 to 0.5, and in our body, from organ to organism, the cell diameter is differ from one organ to the different organs. And in our body, the longest cells are nerve cells are longest cell. In the nature, the largest cells are called as ostrich egg is considered as the largest cell. So these are all are some of the information about the cell biology. They give the foundation, basic foundation to the cell biology discovery. Complexity of organization. Every organism's body is basically made with the cell. So the cell is differ from lower organism to the higher organism during the complexity. Means from the earth starting on, simple organisms are originated. From that simple organisms, the complexity of organization will be occurs during the nature. So how the cellular organization will be occurs? Now we can see. So all the living organisms based on the organization on the earth, they will be divided into mainly two types. One is non-cellular organization, non-cellular organisms. One is called as non-cellular organisms, and another one is called as cellular organisms. One is called as non-cellular organisms. Second one is called as cellular organisms. So means in the nature, the living organisms based on the type of the cell, which type of the cells are present in that organisms, we are divided the organisms into mainly two types. One is called as non-cellular organisms, and second one is called as cellular organisms. So non-cellular organisms means they do not contain particular cell, means the cell having properties are somewhat absent in these non-cellular organisms. Means it is looking like a cell, but the properties or the characteristics which are present in this cell are somewhat absent in these non-cellular organisms. Best example, some viruses. If you see viruses, so viruses are not having particular cell shape, cell structure. So they are having some capsule-like structure which is also made with some protein. And inside this, there will be some genetic material will be present. Inside this, some genetic material. Means the viruses are nothing but nucleoproteinaceous structure. Nucleoproteinaceous structure. So these nucleoproteinaceous structures, they are not having particular cell membrane and particular cell organelles which are present in the cells. But if they are present in the environment, they are acts as non-living things. But if they are present in the living organism's body, they act as living organisms. So these viruses are comes under these non-cellular organisms. Then cellular organisms means whatever the cell organization is present in that particular organisms, they are completely looking like a cell, and the properties or the characteristic features which are applicable by these cells are present in these cellular organisms. For example, if you see the prokaryotic bacteria, or if you see some eukaryotic animals or plants, they are all are containing cells. They are completely, they are acting as real cells. Means the properties, the characteristic features which are present in that cells are completely applicable by these cellular organisms. Again, we are divided the cellular organisms into two types. How many types? Two types. One is called as prokaryotic cells, and the second one is called as eukaryotic cells. One is called as prokaryotic cells, and the second one is called as eukaryotic cells. Prokaryotic cells means, pro means first, karyon means nucleus. So means here these organisms are first formed cells during the evolution. In the evolution, these prokaryotic cells are first formed. And for example, if you see the cell is containing a cell membrane. Inside the cell membrane, there will be another nucleus will be present. The nucleus containing genetic material is surrounded with a particular membranous like structure. So inside that membranous like structure, whatever the genetic material or whatever the hereditary material is present, and surrounding that so many number of cell organelles are also present surrounding this nucleus. So like there will be cell is there, and surrounding the cell there is a cell membrane is there, and cell membrane is inside the cell membrane there are so many number of cell organelles are there. And with that of there will be some nucleus containing genetic material is also present. So which is the main important structure of the cell? Means cell structure commonly we can observe. For example, if you observe in our human beings, or if you observe in some plants, or if you observe in some dogs or foxes, any organism you can observe the cells. Means there is a membrane will be present. Inside this membrane there will be some cell organelles are present, and surrounding these cell organelles a centrally located genetic material will be present. But prokaryotic cells are lack this nucleus containing membrane. Means the genetic material will be present inside this cell membrane, but the cell membrane, inside this cell membrane genetic material is present, but what is lacking in these organisms? The nuclear membrane which is surrounded by this genetic material. The nuclear membrane which is surrounded by this genetic material, and whatever the cell organelles bounding organelles, whatever the membrane bounded cell organelles which are present inside the cell are absent in these prokaryotic cells, but they are present. They are first formed cells. Means here simply we can say that in prokaryotic cells naked nucleus is present. Means the genetic material without nuclear membrane and some of the membrane bounded cell organelles are absent in these prokaryotic cells. For example, you can see the bacteria or blue-green algae and some mycoplasma like organisms, some spirochetes, Rickettsia. So these all the cells are containing the nuclear, all the cells are containing what is that? Prokaryotic in nature. So prokaryotic nature means inside the cell membrane the genetic material is present without nuclear membrane. Means the genetic material is not surrounding with nuclear membrane. Nuclear membrane is somewhat absent. So but the DNA or the genetic material which is present in this prokaryotic cell is present in a particular place. That place is called as nucleoid. In which place it is located? Nucleoid. So in particular place the nuclear membrane, nuclear material or DNA or RNA which is acts as a genetic material for inheritance of the characteristics is present in the particular place. That particular place is contain what is that? Nucleoid. What is that part? Nucleoid. And some of the other membrane bound structures like mitochondria or for example that chloroplast or something different ribosomes are also somewhat absent in this structure. Okay, so some membrane bound organelles are also absent in these prokaryotic cells. Simply prokaryotic cells are first formed cells. In these cells the nucleus is not surrounding with nuclear membrane. There is genetic material will be there, but the genetic material is not surrounding with nuclear membrane, and some membranous bounded organelles are also absent in this structure. And these mitochondria, ribosomes, lysosomes, these structures are also absent in these prokaryotic cells. Then coming to the another type of the structure called as eukaryotic cells. U means true. Karyon means nucleus. Means here during the cell formation, the true nucleus means if you can see the cell structure, there will be some nuclei. This nuclear membrane, the whatever the genetic material which is DNA or RNA is present inside this inside this nucleus, and some of the membrane bounded organelles like mitochondria, chloroplast, Golgi complex, like that cells are also present in this eukaryotic cells. So that's why eukaryotic cells are somewhat advanced in nature. Cells are somewhat primitive or primitive. In eukaryotic cells, the nucleus is surrounded by nuclear membrane, and some membrane bounded cell organelles are present in this structure. The DNA or the genetic material is surrounded by particular nuclear membrane is present. So this is about different cells which are present in the cell. Then coming to the how we can observe these cells. For example, if you see bacteria, how we can observe this bacteria in the laboratory. So the bacteria, the prokaryotic, which are very small, we can't see with our naked eye. After the discovery of this compound microscope and Nolan Ruska's electron microscope, they observed the prokaryotic cells were observed by these microscopes. During the observation of this prokaryotic cells, we are going for a technique called as gram staining technique. So what is the technique? Gram staining technique. So gram staining techniques means we are taking the bacterial culture on the slide, so and we can make a smear. On that smear we can first apply the crystal violet. After the crystal violet application, we can pour some of the grams iodine. After that grams iodine, we can pour some saffronine above that slide. So this crystal violet and saffronine structures, saffronine dyes, we can observe the bacteria. So because the bacteria or the prokaryotic organisms, whatever the bacteria, they are mainly of two types. So Christian Gram, one of the scientists, discovered first time this gram staining technique. In this gram staining technique, they divide this bacteria into mainly two types. One is gram positive bacteria and the second one is called as gram negative bacteria. How can we say that this bacteria is called as gram positive, this bacteria is called as gram negative? So this was we can observe and we can say that this is a gram positive or this is a gram negative by doing a technique called as gram staining technique. In this gram staining we can use three different chemicals. One is called as crystal violet, and second one is grams iodine solution, and third one is called as saffronine. So by using grams iodine, we are observing this bacteria and we are categorized the bacteria into gram positive positive and gram negative. So that's why this name is called as gram staining technique. What is the technique? Gram staining technique. So this is about some of the organizations. Okay, to next class we are going to discuss. Thank you.